This particle, a virus only 100 nanometers in diameter, has plagued our world for nearly 2 years now, causing massive and worldwide fear, death, and disruption on scales unlike most have ever seen. Fortunately, we have also seen a rapid influx of innovation, and companies around the world have developed vaccines to inoculate ourselves against severe sickness. And some of these, such as the Johnson & Johnson vaccine, Russia's Sputnik 5, and the Oxford AstraZeneca vaccine, all utilize a common mechanism for delivery, namely the adenovirus vector. In other words, scientists edit the genome or genetic code for a virus similar to the ones that cause the common cold and insert the SARS-CoV-2 spike code instead of disease-causing genes, which in turn allows our cells to produce antigens against the coronavirus. While I won't be talking about adenoviruses today, I will be discussing a potential virus particle uncovered recently through computational analyses that is related to adenoviruses called Polintons. My name is Sundrani, and I'm a rising senior from Stanford majoring in biomedical computation. I've been working this summer in Dr. Andrew Feyer's lab, and my summer project involves characterizing the evolution and function of these putative particles called Polintons. Before I get into some background, I owe a deep thanks to Dr. Feyer, as well as my mentor, Dr. Daeyoon Jung, the rest of the Feyer lab, and CVI and the Department of Pathology for their immense support throughout the entirety of the summer. To understand what Polintons are, we start with another similar element called transposons, a type of mobile genetic element. These elements are spread throughout our genome as well as in other eukaryotes or multicellular organisms. Thankfully, though, they are mostly inactive because they hold a very special and unintuitive property of transposition, which means they have an ability to jump throughout a genome and can do so in many different ways. This looks like something I detail here, where we have some cell with chromosomes or tightly wound up DNA or genetic code of A's, T's, C's, and G's, where after looking a little bit more closely, we can have a segment that would be a transposon and perhaps another segment that would be a gene of interest. The transposon may then jump, and one example product could look like this, where the result could be a destroying gene, an event that may facilitate evolution over time, and if we can control this jumping, perhaps something that could look like gene therapy. Polintons, on the other hand, were discovered computationally, looking through the genomes of many different eukaryotes and seem to resemble transposons with that same ability to jump from place to place. They're named after a core set of conserved proteins found in all polintons called polymerase, shown here in dark green, and integrase, shown in dark blue, with POL and INT forming the core for their creative name. Interestingly, though, these elements can be as much as 5 times as long as transposons and bear resemblance to viruses like the adenoviruses we use for vaccines in the sense that they all have a sequence of DNA shown here in yellow that seems to code for a viral capsid protein or coat that could form virus particles. Most importantly, though, is that they have never been seen or characterized while jumping, nor does anyone know if they're even functional. All of which represent the key unanswered questions driving my project. So how do we get there? How do we capture a Polinton? I'm interested in 2 pathways, the first being computational and the second being experimental. In the computational search, we're looking for a sequence characteristic starting with the phylum of nematodes, where we look through all publicly available genomes of the nematode phyla and perform a search algorithm to identify potential Polinton candidates. On the other hand, in the experimental approach, we're looking through environmental samples that may or may not contain Polintons to try and capture them in real time, searching for a sequence that can jump into a known piece of DNA, and if so, analyze to see what we found. So far, I've performed a search for Polintons matching all the colored sequence characteristics shown on the left side here. And of the 222 publicly available nematode genome assemblies, I found that 23 of them hold 1 or more Polinton candidates, with just over half of them being in the genus Centrorhabditis. On the right side, I have detailed a few key steps of our ongoing capture experiments involving a positive and negative selection plasmid. So why should you care? And what does this all mean? And why should it matter to you? To start, as we elucidate more and more about Polintons, we can make more hypotheses about the origin of viruses and how they evolve over time. But even more applicable is the fact that we are in a world where new ways to deliver DNA or make changes to a host genome is invaluable. Adenovirus vaccines, while useful for COVID thus far, may not be so in perpetuity, and it's possible to develop resistance against them. If we can discover and eventually engineer the transposition of Polintons, we may provide an alternative mode of DNA delivery that one day could be useful in a fight against the next pandemic, and one day could be used in some form to save your life too. Thank you.